And so it gave us like this quick check that what that self-report was matched up. And I think when you think about self-report data, is there's some things people self-report well and some things they don't.
Hmm.
Pack years of smoking is quite accurate. And apparently caffeine consumption from their coffee is pretty accurate too.
Yeah, yeah.
But dietary factors are not as accurate. Um, but so what we did was that we are looking at all of the peaks on the mass spectrometer. We're not saying we're going to look just at these pesticides. We're looking at all of the peaks. But we're able to identify hundreds of pesticides in these samples. And so we were able to go through and essentially do a pesticide-wide association study. I mean, it was really like an environmental-wide association study, but we had hundreds of pesticides in the analysis. And so by doing that, we could then see, like, of all these different pesticides, which of these were elevated 8 years before they were diagnosed. Now, does that give you causation? Like, can't say that that one caused that, but people's exposures, while they do vary day to day, people tend to live in the same community and have similar occupations over time. And so even if you can only get a snapshot in time, it still tells a lot about what's happening there. And so we're able to do this. We're working on the paper right now. But like we did find some compounds that hadn't previously been connected to it, but there's no confirmation bias here. We were just
Hmm.
looking at whatever
Yeah.
molecules were there. But clearly we need to go back into the laboratory and show, do these chemicals damage certain features? And like in an animal model, we see it, but it allows us to do that complement to that genome-wide association study for a particular disease.
Yeah. So in a study like this, the advantage is you can do things in an unbiased fashion. You don't go in thinking we're going to look for this molecule and this molecule because those are the only ones we know about. You can detect sort of anything.
Exactly.
Yeah.
And even the detection, like a lot of the peaks that we see may not be identifiable. Like we know
Yeah.
exactly what the mass is, but they may not be in a database. And so, like what I like to say, we have a lot of unknowns, but they're not unknowable.
Right.
It's just a matter, like you got to spend time figuring out what they are, but there's ways of determining what they are. But the idea is if you see this peak always showing up in
Yeah.
a certain
Yeah.
condition, then you put that postdoc on it and say, figure out what it
Yeah.
is.
Yeah. Before you do a study like this, there's lots of unknown unknowns. Then after you do it, you've got a lot of known unknowns.
Exactly.
Yeah. Interesting. Okay. So, and obviously, again, these things you're not proving what does or does not cause Parkinson's, but these are very good clues that can then you can then use to go in and do functional tests in the lab in an animal, say.
And then also, if you have genetic data, you can do things like Mendelian randomization on those human studies. I mean, so there's a lot more you can do, but we just have not historically had this systematic and comprehensive analysis of all of these exogenous compounds.
Mm-hmm. Mm-hmm. So when you say the exposome, in my reading of your work, you're talking about the lifetime exposure, everything that someone is exposed to over the course of their whole life. How do you even start to think about how you would track that in humans? Are you doing studies where you're like, these are going to be multi-year long studies and you're taking samples at regular intervals?
So this was something that it seemed daunting when I first heard of this concept. What my approach to this was that if a person goes in to see their doctor because they're ill, the doctor might take a blood sample from them and run a sample on them. So it's like you are who you are the day you're being seen by the doctor. And it's a result of all of your past history. So even if I don't have blood samples from the previous 50 years, what can I measure today? So let's say, for example, you were exposed to some very terrible chemical when you were 8 years old, and it caused these epigenetic changes in you that are changing your biology. There should be evidence of that changed biology when you're at the doctor's office. So you may not see the chemical itself that caused it,
Mm-hmm.
but you might be seeing the disrupted biology that was caused by
Yeah,
it.
you can see a biological echo of a previous exposure.
Yeah. And I think this is like where we have a lot of work to do to kind of piece these things together. But there's a lot of studies that have been conducted where they had samples from people at multiple time points and they're biobanked. And so the idea is that can we go back in and take advantage of those sort of studies to really kind of piece together that life history and also use things like epigenetics and protein modifications and even some of the geospatial data about where they lived and worked and piece those things together. So the sort of life course piece of it is a complicated puzzle that will be hard to pull all those things together. But I don't think you always need to have that entire thing, because if we can show that a person with a certain exposure profile won't respond as well to a drug or have a faster progression of a disease, that can be actionable that day. And so it's like, yes, I'd love to have an annual visit and a blood sample from everybody to piece these things together. We can still work with what we have at this point in time. But we do want to get the sort of life course effect of it. It's just that I think snapshots can still tell you quite a bit.
Hmm. How do you start to think about studying things like exposures in utero, or just an exposure that a woman has while she's pregnant? Part of the reason I bring this up is, these are for both the mother and the developing baby, these are sensitive periods of development. And just to give one point of contact here, I had a podcast that was really interesting with a man named Angel Nadal, and he studies bisphenol A, BPA, microplastics, and other endocrine disruptors. And one of the interesting things he shared with me is these things will disrupt endocrine signaling in anyone. But, if you were to expose someone to some amount of BPA as a healthy adult, it'll basically be some temporary messing with insulin signaling, say. But if you give that same exposure to, say, a pregnant woman, because she's in the sensitive period where her body is rewiring itself to accommodate the baby, those endocrine disrupted effects can get locked into place and stay with her permanently even after the pregnancy ends. And so given that this is such a sensitive period for a pregnant woman or for the developing child, how do we start to think about testing the effects of chemical exposures during those periods of life? Is that something where we really have to focus on animal models, or have you done any work there in humans as well?
So my own lab hasn't done this sort of work, but I have colleagues that do this. Is that there's many studies where they follow pregnant women and they take blood samples from them throughout, through the different trimesters, and they can get cord blood, they can get placental samples, and then follow the child afterwards. And I think those are the types of studies that start getting you those data points. So by measuring the mother's blood sample, you get an idea of their exposures.
Mm-hmm.
And then, like you said, if there's changes in biology, changes in endocrine pathways, those are things that can be measured in the offspring over time. And so I know many people that are working on these sorts of studies, and they're getting that sort of data and getting that cord blood, the placental samples. And so it's to me, it is something you can study this in humans. Like it works, like you can measure these things, and it's just a matter of the right types of samples and like what are those critical windows in there. Again, some animal studies are needed to really figure out some of those parts of it, but those human studies are underway, and I think we're seeing that. We know that high levels of exposures lead to worse, outcomes for the offspring. But pinpointing which ones are the worst, like that's the sort of work we need to do now that we have these tools to look at not just the one chemical, but all of chemical space in those critical periods.
Yeah, and so on the one hand, when we do, there's one type of screen you can do where you sort of know what you're looking for ahead of time, and you have reason to believe, say, a chemical is going to cause some effect. You can go in and look for it. But what you've been talking about is you guys are doing these unbiased screens, so you can kind of catch anything that's there, even if we don't even know what it is yet. Has anything, let's just talk about a particular neurodegenerative disease, I guess. When we think about something like Alzheimer's, are there any particular chemical exposures that we know seem to matter for Alzheimer's disease progression or risk?
So I have multiple studies underway now, where the sort of thing, like what we did, for example, in Parkinson's, we're doing them. I don't have the results yet.
Mm-hmm.
I think there is some evidence that exposure to some organochlorine type things, like the DDT type compounds, we've seen things like elevated DDE in some of the populations. And so when you think about, these are compounds we banned in the 70s, but we can still measure in most people. They're very persistent compounds. And so if you think about people that are now in their 70s or 80s, when those exposures occurred, we do think that some of the very toxic sort of pesticides may contribute to the disease. But for people that are now like 50 or 60, those exposures have been going down. And so
I see.
this is like where I know like the National Institute of Aging has recently funded multiple studies in this space. I